All right, in this video, we'll be looking at translation part two. Translation part two, we're going to go through the steps: initiation, elongation, termination. Uh, Initiation is going to seem like the longest one, mostly because I have some other things to explain under initiation that have to do with translation proper. And so it's going to seem like the longest one, but in general, all these steps are pretty plain. Obviously, if you take this to a deeper level, you're going to discover a lot more. But as far as the AP curriculum is concerned, it's pretty simple as far as the understanding that you all have to know for this. And so again, here's just a picture of the flow of information. I like this picture also. That they have the general flow, right, of DNA to DNA, DNA, RNA, protein, that central dogma. You have some special cases where RNA can be replicated, and you have RNA going to DNA. We talked about that yesterday or in the last video with RNA transcriptase. So again, just general understanding. Make sure when you start to study this stuff that you don't miss the forest for the trees. This is an important understanding. If you don't get this, really the whole unit's kind of a wash. All right, so let's talk about the steps of translation. Again, initiation, elongation, termination. Initiation is going to begin when one of the subunits of the ribosome is going to align itself with the start codon. And so you have the rRNA of the ribosome that is going to align itself perfectly with this section of the messenger RNA called the start codon. Now, what does that even mean? Well, here's another thing for initiation. This actually shows you that really well. There's a start codon here, and this is some other thing. I don't know what that is, but it's okay. You don't need to know what that is either. And here's a tRNA bringing in the first amino acid, which is always going to be this particular amino acid. You don't need to know what that stands for, but it's going to be methionine. Now, what is a codon? Well, a codon is a 3-base unit. AUG is one of those 3-base units. It could be any 3 letters that our RNA is made up of A's, U's, C's, and G's, 4 different letters, and any 3 combinations of that. And so 4 to the 3rd power is 64. And so there are 64 different codons that are possible in this particular thing. And so each one of those codons codes for an amino acid. Here's AUG. It codes for methionine, or it's a start codon. This makes it sound like that methionine is not always the start amino acid. It is. And so how do you use this codon? Well, this codon chart. Well, let's say I have CAU. I just came up with that. Well, C is my first one, so I'm going to come to this C. A is my second one. So C and A meet here, and then CAU right here. It's going to give me histine. I think that's what that one's called. And so, or histidine, one of those two. And so that's going to bring that particular amino acid every single time. And so notice there are 3 codons that do not code for an amino acid. They code for stop. Stop is not the name of an amino acid. It is the name of an action that occurs. When the stop codon is reached, that means stop. That's when termination occurs and the protein is done. All right. And so we need to talk a little bit more about tRNA too, because whereas here's my GAU, let's go back here, GAU, that's ASP. I think that's asparagine maybe. All right. And so this is there it is. Look, it's bringing asparagine every single time. This tRNA only has one job, and it's to go out and get asparagines. How does it know its job? Well, it has this little section down here that matches up perfectly with GAU because it's CUA, which is the anti-codon. It's the opposite of this codon. And so every time as that ribosome is moving along the messenger RNA, it's going to come to these 3-base units, and it's going to bring the appropriate amino acid with it, and this chain is going to get longer. And so let's go back to initiation to make sure that we have everything here. So this initiation, this section of this ribosome is going to line up perfectly with this stop codon. Notice the ribosome has two subunits, not really important for the scope of this class, and it's going to line up perfectly here. It's very similar to you get up and you want to go to school in the morning and it's cold outside and you put on a jacket. And you have to line up that zipper just right or the whole thing is screwed up. And so you have to get that zipper connected just perfectly before you like zip it up, right? And so the same thing is going on here. The ribosome is going to have to align just perfectly. And then once it gets aligned, it is going to basically move down this amino or this messenger RNA sequence like a zipper. And as it moves, it's going to add an amino acid every single codon, and this little chain is going to get longer. This picture I think is better. See, this one's going away. It already brought its amino acid, and it's going off to get another one. Which one is it going to get? Well, it's going to get whatever UGG is. What is UGG? I'm curious. UGG. It's going to go get some more tryptophan, put somebody to sleep. All right. And so here comes another one. tRNA is bringing in this one. It's going, and this is just going to keep happening the whole time until it gets to what? Until it gets to the stop codon, which is termination. Once it gets to the stop codon, there's not, and I like how this one looks like a kite string. Anyway, the stop codon doesn't have a tRNA for it. All right. It has this called release factor, not important. But basically, the release factor, you're going to be surprised to hear this. The release factor causes this polypeptide to release. And then you have this primary structure of protein, right? This long string of amino acids. That protein is going to then fold up a particular way based upon its biochemistry. Those amino acids are going to be attracted to one another in certain ways. It's going to form into a particular shape. That shape is going to have a particular function for that cell in order for that cell to function the way it's supposed to work.